Hi everyone. Welcome to this quiet space, a place where minds slow down and hearts rest. Before we begin this gentle, sleepy journey, just a small moment of support. If this meditation brings you calm, comfort, or clarity, please subscribe to the channel, gently tap the like button, and leave a comment to let us know you are here. Your support helps us continue creating these peaceful spaces for your mind, for your heart. Now, there is nothing else you need to do, nothing to think about. Let's begin. Close your eyes now and slowly settle into a comfortable position. Take a gentle breath in and slowly breathe out. Let your shoulders relax, let your jaw loosen, and allow your whole body to feel heavy. Now, let's gently explore something many of us have felt, even if we've never named it. There is a particular kind of stillness that settles over a forest just before dawn. The air sits heavy and cool. Mist drifts in thin ribbons through the lower branches. Somewhere in the distance, a bird makes a single note and then goes quiet again. And in that pause, in that interval between night and morning, the world seems to hold its breath. Look closely at the ground beneath the trees. Below a thin layer of fallen leaves, below a crust of moss and soil, something rests in the dark. A seed, no larger than a grain of sand, encased in a coat, so thin, you could press it between your fingers, without feeling anything at all. And yet, inside that coat, folded with extraordinary precision, is the entire blueprint of a living organism, roots, and stem, and leaf, waiting. It has perhaps been waiting for months, perhaps for years. There is no impatience in it, no reaching, no straining toward the surface, just stillness and readiness and the slow accumulation of the right conditions. Above that seed, the forest is already alive. With quiet motion, water is rising through the interior of every trunk, drawn upward against gravity, through channels so narrow you could not see them. With the unaided eye, leaves are turning their surfaces toward the increasing light, adjusting the tiny openings on their undersides in response to humidity and temperature. Sugars produced yesterday are still moving through tissues, from leaf to root, feeding cells. Along the way, the forest is not asleep, it is simply working in a register, too slow and too small for us to notice. At a glance, this is the central quality of plant life that we so often overlook. We say that plants are still. We say they do not move, do not respond, do not react. But this is only true at the speed we happen to live at. Slow the world down. Slow it down by hours, by days, by seasons. And what you see is extraordinary. Roots navigating through rock and clay, sensing moisture and mineral content, branching where conditions improve, withdrawing where they do not, stems orienting toward light with a precision that would be impressive in any machine, flowers opening and closing in response to temperature, to the presence of insects, to the angle of the sun. Plants are not passive. They're simply patient, and in their patience, they have built the living world. Every forest you have ever walked through, every meadow you have ever crossed, every garden and hedgerow and coastal cliff, thick with grasses, all of it is the accumulated work of organisms that have been quietly engineering the land for nearly 500 million years. The oxygen in the air you breathe right now was produced by plants. The soil beneath your feet was shaped, in large part, by plant roots and plant decay. The food that sustains your body traces back, in almost every case, to a plant that converted sunlight into chemical energy and stored it in a form that could be eaten. We live, quite literally, inside the work that plants have done, and yet, most of us walk past them without looking. This documentary is an invitation to look more carefully, to move at a slower pace through the science of plants, to sit with the ideas long enough for them to become clear. This is Botany, offered as simple science explanations for beginners. No jargon left unexplained, no concept rushed past, no detail treated as too small to be interesting, because in botany, the small details are often the most astonishing ones. We will begin at the beginning with what plants are and how scientists have come to understand them. We will travel through the history of plant life on Earth, from the first green films on ancient rocks to the forests and grasslands and wetlands of today. We will look inside a single plant, at its roots and stems and leaves, and follow the movement of water and light and sugar through its interior. We will examine flowers and seeds, pollination and germination, and the remarkable ways that plants move through the world without moving at all. And then we will step back and look at the larger picture, at the role of plants in ecosystems, in human civilization, in the ongoing work of science. We will look at what researchers are learning about plants today, and at the quiet complexity that continues to emerge from laboratories and field stations around the world. There is no hurry. The forest is not going anywhere. The seed in the ground is patient. So let us begin slowly, as all good things in botany begin, with stillness, with attention, with the long, careful, unhurried work of looking. Botany is the branch of biology devoted to the study of plants. It is one of the oldest formal sciences humanity has practiced, older than chemistry, older than physics, as we know it today, older in many ways than the idea of science itself. People have always needed to know plants, which ones were safe to eat, which ones could ease a fever or close a wound, which ones would destroy a crop, and which ones would yield it. Long before anyone wrote down a definition of botany, people everywhere were making careful observations about the green world around them, accumulating knowledge across generations, building up libraries of understanding, stored not in books, but in the practices and traditions of communities. The formal study of plants, what we now call botany, began to take shape in ancient Greece, where thinkers like Theophrastus wrote careful descriptions of plant forms and habits. Over the centuries that followed, the work of understanding plants deepened and widened. By the time of the great age of exploration, botanists were traveling the world to collect and catalog species. By the 19th century, the development of the microscope had opened up a completely new level of plant anatomy, revealing cells and tissues and structures invisible to the naked eye. And in the 20th century, the development of molecular biology gave botanists tools to examine the genetic instructions inside plant cells, the code that determines how a seedling becomes a tree, how a flower knows when to open, how a root knows which direction to grow. Today, botany is a vast field with many branches. There are plant anatomists who study plant structure, plant physiologists who study plant function, plant ecologists who study how plants interact with their environments, plant geneticists who study plant DNA, plant taxonomists who classify and name the extraordinary diversity of plant species, and paleobotanists who study the fossil record of ancient plant life. Each of these specialties contributes to a collective picture of plants that continues to grow, more detailed and more surprising with each passing decade. But before we can explore any of those branches, we need to answer a more basic question. What, exactly, is a plant? This turns out to be a more interesting question than it might first appear. In everyday language, we use the word plant loosely. We might say that algae is a plant, or that a mushroom is a plant, or that bacteria growing on a rock are a kind of plant. But in the careful language of biology, the word plant has a more specific meaning. True plants, the organisms that botanists study, are a group of multicellular organisms that share a set of fundamental characteristics. They are made of cells, enclosed in rigid walls, composed largely of cellulose, a fibrous material that gives plant cells both structure and protection. They are capable of photosynthesis, the process by which they use the energy of sunlight to build organic molecules from water and carbon dioxide. They reproduce through a characteristic life cycle that involves alternating generations, and they are descended from a shared ancestor, a group of freshwater algae that hundreds of millions of years ago gave rise to the lineage that would eventually colonize the land. Algae, despite their green color and their capacity for photosynthesis, are not considered true plants by most modern botanists. Many algae are single-celled organisms, and even the more complex, multicellular algae lack some of the defining features of land plants. They are, however, closely related to plants, and the story of how some ancient algae gave rise to the plant kingdom is one of the most consequential chapters in the history of life on Earth. Fungi, mushrooms, and molds, and yeasts are not plants at all, despite the fact that they grow from the ground and can be mistaken for plants. At a glance, fungi are more closely related to animals than they are to plants. They do not photosynthesize. They obtain their energy by breaking down organic material, dead wood, fallen leaves, the tissues of other organisms. They have their own kingdom, distinct and fascinating, and they interact with plants in ways we will explore later. In this documentary, bacteria are even further removed from plants. These are single-celled organisms, without even the kind of cell nucleus that plant cells possess. Many bacteria can photosynthesize. The earliest photosynthetic life on Earth was bacterial, but they are not plants in any sense of the word. The plant kingdom itself is thought to include somewhere between 300,000 and 400,000 described species, with estimates suggesting there may be many more still undiscovered. This diversity ranges from microscopic mosses growing on wet stone to towering redwoods more than 100 meters tall. It includes flowering plants and conifers, ferns and horsetails, liverworts and mosses, organisms that have evolved an extraordinary range of forms and strategies for living. What unites all of these organisms, what makes them all plants, is not their appearance, but their underlying biology, the way they build their cells, the way they conduct photosynthesis, the way they reproduce, the way they manage water. These shared characteristics are the signature of a shared ancestry, a lineage that stretches back nearly 500 million years to the first green plants on land, and running through all of it, through every cell, every leaf, every root, is the fundamental process that makes plants what they are, the conversion of sunlight into life. Simple science explanations for beginners often describe this as plants eating sunlight, and while that is a simplification, it captures something true. Plants are, above all else, light catchers. everything else they do, everything that makes them remarkable, is built on that foundation. To understand where plants come from, we have to travel back to a world that would be almost unrecognizable to us today. Imagine the Earth as it was roughly 500 million years ago. The planet itself is ancient by this point, already 4 billion years old, but the land surface is still largely bare. Rock and sand and dust, shaped by wind and rain and the slow grinding of geological time. The atmosphere contains oxygen, but in different proportions than today. The land has no soil as we would recognize it, no dark, rich earth, full of organic matter, just weathered mineral material, accumulating slowly on ancient shorelines and continental interiors. But the seas are alive in the shallow coastal waters, in tidal pools and estuaries, and the lit upper layers of ancient oceans. Photosynthetic organisms have been thriving for billions of years. Cyanobacteria, microscopic, ancient, and extraordinarily successful, have been producing oxygen and organic matter since long before anything with a cell nucleus existed. More recently, in evolutionary terms, a different kind of photosynthetic organism had arisen. The algae. These organisms had a more complex cellular structure than bacteria, with a nucleus and specialized internal compartments, and many had developed multicellular bodies of considerable complexity. Among the algae, living in shallow freshwaters, possibly in tidal zones, where freshwater and seawater met, were organisms that would eventually give rise to the land plants. Current research suggests that the closest living relatives of land plants are a group of green algae called the charophytes, which today include organisms like stoneworts growing in clear lakes and ponds. The evidence for this relationship comes from shared biochemical features, similar cell division mechanisms, and molecular comparisons between charophyte DNA and land plant DNA. The transition from aquatic algae to land plant was not a single dramatic event. It was a gradual accumulation of adaptations over millions of years, each small change providing some benefit, some slight advantage, in the increasingly challenging environment of the land surface. What were the challenges of life on land? They were considerable. Water, in an aquatic environment, is everywhere. An alga, floating in a pond, never has to worry about drying out, but exposed to air. An organism faces constant desiccation. Water evaporates from surfaces. The sun, without the filter of water, delivers intense radiation. There is no medium to carry away waste products, and gravity, so easily managed when you are suspended in water, becomes a constant force that any structure rising above the ground must be built to resist. The first land plants, or pre-plants, appear to have been small, flat, low-growing organisms that stayed close to moist surfaces, somewhat like modern liverworts. Fossil evidence from roughly 470 million years ago shows the presence of simple plant-like organisms that lacked complex internal structures but could survive brief exposures to air. Current evidence indicates that these early organisms were able to tolerate drying and then rehydrate when moisture returned, a strategy still employed by some modern mosses and liverworts today. Over the following tens of millions of years, the lineage of land plants gradually acquired a set of adaptations that allowed them to move further from water and to grow larger and more complex. One of the most critical of these adaptations was the development of a waterproof outer layer, the cuticle, covering the surfaces of leaves, and stems, this waxy coating, dramatically reduced water loss from plant surfaces, allowing plants to retain moisture even in drier conditions. But it created a new problem. If the surface is sealed, how does the plant exchange gases with the atmosphere? Photosynthesis requires carbon dioxide to come in and produces oxygen that must go out. The solution was the development of stomata, tiny adjustable pores in the leaf surface that can open to allow gas exchange and close to prevent water loss. Another transformation was the development of vascular tissue, internal channels capable of conducting water and dissolved nutrients from one part of the plant to another. Early plants, without vascular tissue, were limited in size. Water could only diffuse slowly through their tissues, limiting how tall or how complex they could grow. The appearance of vascular plants, sometime around 400 million years ago, opened up an entirely new dimension of growth. With internal plumbing, plants could grow taller, reaching more light. They could develop roots to anchor themselves and to explore larger volumes of soil. They could build woody stems capable of supporting great weight. The development of roots was itself a transformative event, not just for plants, but for the land surface itself. Roots penetrating rock and soil accelerated the weathering of minerals. Root decay added organic matter to mineral substrate, beginning the long process of soil formation. The presence of roots changed the hydrology of the land surface, anchoring soil against erosion and altering the way rainwater moved through the ground. When plants arrived on land, they did not simply occupy an existing environment. They began, slowly and invisibly, to create the environment that all land life would eventually depend on. By roughly 360 million years ago, during a period geologists call the Devonian, the land surfaces of the ancient world had been fundamentally transformed. Forests, real forests, with trees tens of meters tall, had appeared. The earliest forests were composed of unfamiliar species, nothing quite like what grows today. Tree-sized lycopsids and horsetails and early seed plants. But forests, they were, and with them came soils and watersheds and the first ecological communities of the land. Scientists believe that the oxygen content of the atmosphere rose significantly during this period of rapid plant colonization, as the massive increase in photosynthetic life produced oxygen faster than it could be consumed. The atmospheric changes associated with early plant colonization had effects that reached far beyond the plant world. They helped shape the conditions that allowed land animals, including the ancestors of all terrestrial vertebrates, to eventually emerge and diversify. The story of how plants came onto land is, in a very real sense, the story of how the world we know became possible. Hold a plant in your hands for a moment, a simple one, perhaps a small wildflower, pulled gently from a garden bed. Look at it carefully. You are holding several distinct systems, each one specialized for a particular task, each one connected to the others in a web of dependence and exchange. The roots, the stem, the leaves, the flower, if one is present, and if the season is right, the seeds. Each of these structures tells a story about how plants have solved the particular problems of living on land. Let us begin at the bottom, in the soil, where the roots are. The root system of even a small plant is more extensive than it appears. What you see when you pull a plant from the ground is often only a fraction of the actual root structure. The larger roots that were near enough to the surface to come free. A plant growing in undisturbed soil will typically have a root system that penetrates the soil in many directions, branching repeatedly, filling the available space with a network of channels and surfaces. In some plants, the root system occupies a larger volume of space than the above-ground parts. Roots have two primary jobs: anchoring the plant in place and absorbing water and dissolved minerals from the soil. They also serve as storage organs. In many species, the carrot you eat is largely a swollen root, packed with sugars and starches, and, as we will explore more fully in a later section, roots interact with the surrounding soil in ways that extend far beyond simple absorption. Moving upward from the roots, we encounter the stem. The stem is the structural backbone of most plants, the central axis that holds everything else in place and provides the conduits through which materials move. In small, soft-bodied plants, the stem is green and flexible. Its cells full of water that gives it rigidity. In woody plants, the stem is transformed over time by the addition of harder material, wood, that allows it to grow tall and to support the increasing weight of branches, leaves, and fruit. The stem connects roots to leaves, and in doing so, serves as the highway for two of the most important flows within a plant. Water and minerals, absorbed by the roots, travel upward through the stem to the leaves and flowers above. Sugars produced in the leaves through photosynthesis travel in the other direction, downward through a different set of channels to reach the roots, the growing tips, and other parts of the plant that need energy. At the growing tips of the stem and at the tips of every branch, there are zones of actively dividing cells called meristems. These are the plant's growth points, the places where new cells are produced, where new leaves and flowers and branches are added to the plant body. A meristem at the tip of a shoot is called the apical meristem, and it is responsible for the elongation of that shoot. Understanding meristems is central to understanding how plants grow, because unlike animals, plants do not grow by enlarging an already formed body. They grow by continuously adding new tissue at their tips and margins. Now look at the leaves. These flat, thin structures are, in most plants, the primary sites of photosynthesis, the engines that capture sunlight and convert it into chemical energy. Their flattened shape maximizes the surface area available to catch light. Their green color comes from the presence of chlorophyll, the pigment that absorbs light energy, and their interior is organized with considerable precision to carry out the chemistry of photosynthesis as efficiently as possible. A typical leaf consists of an outer layer of tightly packed cells, the epidermis, that protects the interior and limits water loss. Beneath this is a layer of loosely packed, chloroplast-rich cells, where most of the photosynthesis happens. Running through the leaf are veins, the leaf's vascular system, which deliver water and carry away the sugars the leaf produces. And on the lower surface of most leaves, scattered among the other epidermal cells, are the stomata, those adjustable pores through which the leaf breathes. In flowering plants, the dominant group in today's world, the stem may also bear flowers. The flower is the plant's reproductive organ, and it is among the most elaborately specialized structures. In the plant kingdom, a typical flower consists of petals, which attract pollinators, sepals, which protect the developing bud, stamens, which produce pollen, and the pistil, which contains the ovules that will develop into seeds. If fertilized, the extraordinary diversity of flower forms across the plant kingdom reflects the extraordinary diversity of the pollinators and environmental conditions to which flowering plants have adapted over their evolutionary history. Finally, from fertilized flowers come seeds. A seed is a remarkable object, a complete plant embryo with a small supply of food to sustain it through germination, all enclosed in a protective coat. Seeds are the mechanism by which flowering plants and conifers disperse through the environment, and the diversity of seed forms reflects the diversity of dispersal strategies plants have evolved. Some seeds are dust-like and travel on the wind. Others are encased in fleshy fruit and travel inside animals. Still others have hooks that catch on fur. The seed is, in many ways, the most important single innovation in the history of land plants, and we will give it the attention it deserves in a later section. For now, hold that image of a complete plant: roots, stem, leaves, flower, seed, and think of it not as a fixed object, but as a set of processes: water flowing upward, sugars flowing downward, light being captured and transformed, gas being exchanged through microscopic pores, growth happening cell by cell at 100 growing tips simultaneously. A plant is not a thing so much as it is a set of movements running continuously in every direction at once. On a clear morning, when the sun has risen just high enough to clear the tree-line, there is a moment when the angle of the light is exactly right, when it passes through the upper canopy at a shallow angle and seems to illuminate individual leaves from within, turning them into small green lanterns suspended in the air. That light is doing something, not just illuminating, not just warming. It is being absorbed by structures inside those leaves, and it is being used to do chemistry. Photosynthesis is the process by which plants and certain other organisms convert the energy of light into the energy stored in chemical bonds. It is the foundation of almost all food webs on Earth, the source of the oxygen in our atmosphere, and the mechanism by which the sun's energy is made available to the rest of the living world. Understanding it is perhaps the most important single task in understanding plants. Let us begin with the basics. A plant leaf takes in three things from its environment to carry out photosynthesis: sunlight, carbon dioxide, and water. From these three inputs, it produces two outputs: oxygen, which is released into the atmosphere, and glucose, a simple sugar that the plant uses as a source of energy and as a raw material for building other molecules. The simplified summary of photosynthesis can be written as carbon dioxide plus water in the presence of light yields glucose plus oxygen. But this summary, while accurate as far as it goes, conceals an enormously complex series of chemical reactions occurring inside specialized structures within plant cells. Those structures are called chloroplasts. Each chloroplast is a small, lens-shaped organelle, a membrane-enclosed compartment within a plant cell. A single leaf cell may contain dozens of chloroplasts, and a single leaf may contain millions of cells. The interior of a chloroplast is organized into a system of stacked, disc-like membranes called thylakoids, surrounded by a fluid called the stroma. The thylakoid membranes are where light is captured. Embedded within these membranes are clusters of pigment molecules. Most importantly, chlorophyll. Chlorophyll is a remarkable molecule because of the way it interacts with light. It absorbs light strongly in the red and blue portions of the visible spectrum and reflects light in the green portion. This is why plants look green. To our eyes, we are seeing the wavelengths of light that chlorophyll does not use reflected back to us. When a photon of light strikes a chlorophyll molecule, its energy is absorbed and transferred to electrons within the molecule, raising them to a higher energy state. These energized electrons are then passed along a chain of molecules embedded in the thylakoid membrane, a process that releases energy in controlled steps and uses that energy to pump hydrogen ions across the membrane. The flow of hydrogen ions back through a specialized protein complex drives the production of ADP, the cell's universal energy currency. This part of photosynthesis, the light-dependent reactions, is, in essence, a highly efficient biological solar panel, converting light energy into chemical energy. The other part of photosynthesis, called the Calvin cycle, or the light-independent reactions, takes place in the stroma of the chloroplast. Here, the ADP and another energy-carrying molecule produced in the light reactions are used to drive a cycle of chemical reactions that take carbon dioxide from the air and incorporate it into organic molecules. The final product of this cycle, after several steps, is a 3-carbon sugar that the plant can use to build glucose, starch, cellulose, and all the other organic molecules it needs. This incorporation of carbon dioxide from the air into organic molecules is called carbon fixation, and it is one of the most consequential chemical processes on Earth. Every carbon atom in every plant, in the wood of a tree, in the grain of a wheat plant, in the skin of an apple, was once a carbon dioxide molecule floating in the atmosphere. It was pulled out of the air, step by step, through the chemistry of photosynthesis, and assembled into the fabric of a living organism. The oxygen released by photosynthesis is a byproduct of the process. In the light reactions, water molecules are split apart to provide the electrons needed to replace those lost from chlorophyll. When water is split in this way, oxygen is released as a byproduct, and this oxygen, molecule by molecule, is vented through the stomata of the leaf into the surrounding atmosphere. The oxygen-rich atmosphere we breathe today is the cumulative product of billions of years of photosynthesis, first by ancient cyanobacteria, and later by land plants and algae. Different plants have evolved variations on the basic photosynthetic chemistry to cope with different environments. In hot, dry conditions, the standard photosynthetic pathway loses efficiency because stomata must be kept closed for much of the day to prevent water loss, and closed stomata mean reduced carbon dioxide availability inside the leaf. Some plants in these environments, including corn and sugarcane and many grasses native to warm climates, use a modified pathway called C4 photosynthesis that concentrates carbon dioxide in specialized cells reducing the problem of low availability and even more extreme modification found in many succulent plants adapted to desert conditions allows the plant to open its stomata only at night when temperatures are cooler and water loss is reduced, storing carbon dioxide in chemical form until daylight provides the energy to use it. These variations on the photosynthetic theme are elegant examples of how evolution has fine-tuned a fundamental process to suit an extraordinary range of environments. The basic chemistry is conserved. The details have been modified by natural selection over millions of years to fit the particular challenges each group of plants faces in any form, in any environment. Photosynthesis is the same thing: the capture of sunlight, the transformation of simple molecules into complex ones, the quiet, continuous, invisible work of turning energy into life. If you have ever tried to remove a mature weed from a garden, one of those plants with deep, fibrous roots that seem to extend far beyond where any reasonable root should go, you will have a sense of the tenacity of plant root systems. The visible part of a plant is often just half the story, sometimes less than half, below the soil surface, extending through the dark earth. In every direction is a structure of comparable complexity and often comparable size to everything above ground. Roots are the hidden architecture of the plant world, and they are doing work that is both essential and surprisingly complex. The primary task most people associate with roots is the absorption of water. This is indeed a critical function. Plants require enormous quantities of water. Most of the water absorbed by roots eventually evaporates from leaf surfaces in a process called transpiration, which, as we will see, plays an important role in the movement of water through the plant. But roots do not absorb water evenly across their entire surface. Water absorption is concentrated in a zone near the growing tip of each root, where specialized cells, called root hair cells, are found. Root hairs are remarkably simple structures. They are just extensions of individual epidermal cells on the root surface, elongated into thin filaments that project into the surrounding soil. But they are enormously effective at what they do. Root hairs increase the surface area of the root dramatically. A single plant may have billions of root hairs, and they penetrate the spaces between soil particles, coming into direct contact with the water films that cling to those particles. Water moves into root hairs by osmosis. The concentration of dissolved substances inside root hair cells is higher than in the surrounding soil water, which means water tends to flow inward across the cell membrane. Once inside the root, water moves inward toward the center through a series of cell layers, and from there into the vascular tissue that carries it upward through the plant. Alongside water absorption, roots also take up mineral nutrients from the soil: nitrogen, phosphorus, potassium, calcium, iron, and a range of other elements that plants need in smaller quantities. These minerals, dissolved in soil water, enter root cells through a variety of transport mechanisms, including specialized protein channels in cell membranes that are selective for specific ions. The acquisition of mineral nutrients is not always straightforward. In many soils, some essential minerals are present in low concentrations or in chemical forms that plants cannot directly absorb. Here, the interactions between roots and the living community of the soil become important. Research suggests that the majority of land plants form close associations with soil fungi in a relationship called mycorrhiza, a Greek-derived word meaning fungus root. In these associations, fungal threads, called hyphae, grow in intimate contact with root cells, and the two organisms exchange resources. The plant provides the fungus with sugars produced by photosynthesis, the fungus with its extensive network of fine filaments that penetrate the soil far more thoroughly than roots alone can manage, provides the plant with mineral nutrients, particularly phosphorus, that might otherwise be difficult to access. The mycorrhizal network, associated with a single tree, can extend many meters through the soil, threading through the root zones of neighboring plants and connecting them all into a shared fungal web. Scientists have documented the movement of nutrients and even carbon compounds through these networks between different plants. The exact significance of these transfers, how much they matter to individual plants under normal conditions, is a topic of ongoing research, and scientists continue to refine their understanding of how these relationships function in real ecosystems. What is clear is that mycorrhizal associations are extraordinarily common, and that they appear to benefit the growth and survival of plants in many environments, particularly nutrient-poor ones. Roots also interact with the soil in more mechanical ways. As roots grow through soil, they push particles aside and create channels through which water and air can move. The exudates that roots release, a mix of sugars, amino acids, and other organic compounds, feed communities of bacteria and fungi that live in the immediate vicinity of the root surface, a zone called the rhizosphere. The composition of these microbial communities differs from the soil further away and appears to be influenced by chemical signals from the plant. Research suggests that this rhizosphere community plays roles in plant nutrient availability and protection against disease, though the details of these interactions are still being actively studied. Different plants develop different types of root systems suited to their particular environments and needs. A taproot system, like that of a carrot or a dandelion, has one dominant central root that grows deeply into the soil with smaller lateral roots branching from it. A fibrous root system, more common in grasses and many other plants, consists of many roots of roughly equal size, spreading outward from the base of the stem, forming a dense, shallow network. Each type has advantages. Taproots can reach water deep in the soil during drought, while fibrous roots spread across a large volume of soil, anchoring the plant firmly and capturing nutrients near the surface. The roots of some plants have been modified for other purposes altogether. Aerial roots in tropical epiphytes, plants that grow on other plants without parasitizing them, absorb moisture from the air. Prop roots in corn plants emerge from the lower stem and grow down into the soil, providing additional structural support. The roots of mangrove trees, growing in oxygen-poor, waterlogged soils, send specialized branches upward into the air to facilitate gas exchange. Even the plants we pass every day have root systems adapted to their particular circumstances in ways that repay a moment's careful attention beneath the surface of any patch of ground. The soil is not empty space. It is full of life, and architecture, roots navigating through darkness, following gradients of moisture and mineral concentration, building structures that will persist long after the visible plant above them has gone. Imagine, for a moment, the problem a tall tree faces. On a summer morning, its leaves are 40 meters above the ground, catching sunlight, opening their stomata, beginning to photosynthesize. As the morning warms, the leaves begin to lose water vapor through those open stomata, and to replace this water, they need more to arrive from below. Meanwhile, the roots in the soil, 40 meters down, are absorbing water steadily, and the sugars produced by those high leaves need to travel back down to the roots, the trunk, and all the growing points along the way. How does any of this happen? How does water rise 40 meters against gravity in large enough quantities to supply millions of actively photosynthesizing leaves? And how do dissolved sugars flow in the opposite direction simultaneously through the same stem? The answer lies in the plant's vascular system, a set of specialized tissues that run through stems, branches, and roots like internal highways, carrying different materials in different directions. Plant vascular tissue comes in two types. The first is called xylem, and it is responsible for carrying water and dissolved minerals upward from the roots to the rest of the plant. The second is called phloem, and it is responsible for carrying dissolved sugars and other organic molecules from the leaves, where they are produced, to the growing tips, roots, and other tissues that need them. Xylem is, in a literal sense, the harder of the two tissues. Xylem cells become woody as they mature. They lay down reinforced cell walls, and then, in many cases, die, leaving behind hollow tubes through which water can flow. These hollow tubes, stacked end to end through the plant, form the conduits of the water conducting system. In trees, the accumulation of old, non-conducting xylem in the center of the trunk forms what we call wood, the dense, strong material that allows trees to grow tall without collapsing under their own weight. The mechanism by which water rises through xylem has fascinated scientists for centuries. The driving force is not a pump. Plants have no heart, no muscle, no mechanical device to push water upward. Instead, the movement of water through xylem is driven largely by transpiration, the evaporation of water from leaf surfaces. As water evaporates from leaves through the stomata, it is replaced by water from the cells beneath, which in turn draw water from the xylem vessels in the leaf veins. This creates a tension, a slight pulling force, in the water column within the xylem, because water molecules are strongly attracted to each other through a property called cohesion, and because they also adhere strongly to the walls of the xylem tubes through a property called adhesion. This tension is transmitted downward through the entire water column, all the way to the roots. The result is that water is, in effect, pulled upward through the plant, not pushed from below. This mechanism, known as the cohesion tension theory, was worked out by scientists in the late 19th and early 20th centuries, and it remains the accepted explanation for water transport. In tall plants, it works because water is an unusually cohesive liquid. Its molecules hold together with enough strength to sustain the tension required to lift water columns many meters high. Engineers have found it difficult to design artificial systems that replicate this feat. using the same principles. The phloem system operates differently. Phloem cells are alive, at maturity, connected to each other through pores in their shared walls, and they transport dissolved sugars and other organic molecules through a process driven by differences in pressure at sites where sugar is being loaded into the phloem, primarily in the leaves. Water is drawn in by osmosis, raising the pressure in those sections of the phloem at sites where sugar is being removed. In roots, growing tips, or storage organs, water exits and pressure falls. This pressure gradient drives the movement of phloem contents from high-pressure regions to low-pressure ones through the living tubes of the phloem network. The spatial arrangement of xylem and phloem within a stem is not random. In most flowering plants, bundles of xylem and phloem are organized together in vascular bundles, arranged in a characteristic pattern, either in a ring around the center of the stem in dicotyledonous plants, or scattered throughout the stem in monocotyledonous plants. This distinction in vascular bundle arrangement is one of the features botanists use to classify flowering plants into these two major groups. In addition to its role in transport, the stem provides structural support and houses the growth zones from which new leaves, branches, and flowers develop. The growing tips of stems contain meristematic tissue, zones of actively dividing cells, and along the stem, at the bases of leaf stalks, there are lateral buds that can give rise to new branches. The arrangement of leaves and branches along a stem is not random. It follows precise mathematical patterns that optimize the way leaves are distributed in three-dimensional space, minimizing shading of one leaf by another. Walk slowly past any hedge, any woodland edge, any forest path, and look at the stems of the plants you pass. The green, wiry stems of climbers, the smooth gray bark of young beeches, the corky ridged surface of an old oak, the hollow, jointed stems of grasses and bamboos. Each of these reflects a particular solution to the challenges of structural support, water transport, and growth in a particular environment. The stem is not just a stalk, it is a precisely engineered conduit, support structure, and growth system, all at once. On a still August afternoon, when the sun is high and the air is warm and thick with the smell of grass and earth, find a single leaf on a tree and look at it carefully. It is thin, barely a fraction of a millimeter from surface to surface. It is flat, presenting as much surface area as possible to the overhead sun. It is green, with chlorophyll, and it is doing something quite extraordinary. It is capturing photons of light and using their energy to rearrange atoms into new molecules. The leaf is the primary site of photosynthesis in most plants, and its structure is organized with considerable elegance. To carry out this task efficiently, let us follow a beam of light as it enters a leaf. The first thing the light encounters is the epidermis, a single layer of transparent, tightly packed cells that forms the outer skin of the leaf. These cells are largely transparent, allowing light to pass through, and they are covered with a waxy cuticle that reduces water loss scattered among them. Mostly on the lower surface of the leaf are pairs of specialized guard cells that form stomata, the adjustable pores through which the leaf breathes. Passing through the upper epidermis, light enters the mesophyll, the interior of the leaf, where photosynthesis actually occurs, the upper portion of the mesophyll, directly beneath the epidermis, is composed of densely packed, elongated cells called palisade mesophyll cells. These cells are particularly rich in chloroplasts, and they are oriented vertically in columns, which maximizes the amount of light that can be absorbed by their chloroplast-packed interiors. The palisade layer is where the majority of photosynthesis in most leaves takes place. Below the palisade layer is the spongy mesophyll, a more loosely organized region of cells with large air spaces. Between them, these air spaces are connected to the outside through the stomata and serve as an internal atmosphere for the leaf, allowing carbon dioxide to diffuse inward from the stomata to the photosynthesizing cells and allowing oxygen to diffuse in the opposite direction, running through. All of this are the veins, bundles of xylem and phloem that deliver water to the mesophyll cells and carry away the sugars they produce. In most flowering plant leaves, veins branch repeatedly from a central midrib, creating a network that reaches every part of the leaf. This venation pattern is one of the features used to identify different plant species, and it ensures that no cell in the leaf interior is far from a vein, which means no cell is far from its water supply. The stomata deserve particular attention because they sit at a crucial intersection. Through stomata, the leaf takes in carbon dioxide, the raw material for photosynthesis. Through the same pores, it loses water vapor, an inevitable consequence of opening up to the atmosphere. Managing this trade-off is one of the central challenges of plant physiology, and the stomata are the mechanism through which it is managed. Each stoma is formed by two guard cells, banana-shaped cells with thickened inner walls that sit facing each other across a pore. When the guard cells are turgid, full of water, and pressing outward, the thickened inner walls curve, pulling the cells apart and opening the pore. When the guard cells lose water and become flaccid, they relax, and the pore closes. The opening and closing of stomata is controlled by a variety of signals, including light intensity, carbon dioxide concentration inside the leaf, humidity, and even chemical signals associated with water stress. Under ideal conditions, plenty of water available, moderate temperatures, high light, stomata open wide, and the leaf carries out photosynthesis at maximum rate. Under hot, dry conditions, when water stress threatens, stomata close to prevent further water loss, even if this means reducing photosynthesis. The plant, in effect, must constantly make a trade-off between maximizing photosynthetic productivity and minimizing water loss. The sensitivity of stomata to environmental conditions allows the plant to adjust this balance moment to moment. Leaves come in an astonishing variety of shapes and sizes, and this variety reflects the diversity of environments plants have occupied. Broad, flat leaves in forest understories maximize light capture in the dim conditions beneath the canopy. Narrow, needle-like leaves in conifers reduce surface area and water loss. In cold, dry conditions, thick, succulent leaves in desert plants store water and have reduced surface-to-volume ratios that minimize evaporation. Hairy leaf surfaces in many species reduce wind exposure and create a layer of still air near the surface that slows water loss. Even the arrangement of leaves on a stem called phyllotaxis, is not arbitrary. In most plants, leaves are arranged in spirals or alternating patterns that minimize the degree to which any one leaf shades the ones beneath it, maximizing the overall light-capturing capacity of the plant's canopy. The mathematical precision of these patterns has been noted since antiquity and continues to be studied by both botanists and mathematicians. A forest canopy, seen from above on a summer day, is essentially a vast collective solar array, millions of individual leaves, each one a precisely engineered light-capturing surface, arranged to intercept as much of the incoming sunlight as possible. The efficiency of this arrangement is part of what makes photosynthesis so effective at capturing the sun's energy and making it available to the rest of the living world. There is a particular kind of morning in late spring when a meadow seems to have changed overnight, when what was the evening before a field of green grass and undistinguished foliage has become, by morning, a scatter of color, yellow and white and blue and purple, appearing as if the field itself decided, sometime in the night, to announce itself. Flowers are, in the most immediate sense, beautiful, but they are not beautiful by accident, and they are not beautiful for our benefit. The petals, the colors, the shapes, the fragrances, all of them are solutions to a single biological problem: how to move pollen from one plant to another. Flowering plants, technically called angiosperms, first appeared in the fossil record roughly 130 to 150 million years ago. Though research continues to refine these estimates, their appearance was a major event in the history of plant life. Within a relatively short geological time span, they diversified into what is now the dominant group of land plants, comprising the vast majority of plant species alive today. Much of this success is thought to be connected to the evolutionary partnership between flowering plants and the animals that pollinate them. To understand flowers, we need to understand plant reproduction. Plants, like animals, reproduce sexually by combining genetic material from two different individuals. In plants, the male genetic contribution is carried in pollen, tiny grains produced in structures called anthers, which sit at the top of the stamens, the male reproductive organs of a flower. The female contribution is housed in the ovule, which sits inside the ovary, the base of the pistil. The female reproductive organ for fertilization to occur. Pollen from one plant must reach the pistil of another plant of the same species. This is the challenge that flowers solve. A plant cannot walk to another plant and hand over its pollen directly. It needs an intermediary, and the flower is the advertisement, and in many cases, the reward for the services of that intermediary. The most common intermediaries are insects. Bees, in particular, are extraordinarily important pollinators for a vast range of flowering plant species. A bee visiting a flower in search of nectar, a sugary liquid secreted by glands at the base of the petals, brushes against the anthers and picks up a dusting of pollen on its body. When it visits the next flower of the same species, seeking more nectar, some of this pollen is transferred to the sticky surface of the pistil, the stigma, where it can germinate and eventually fertilize the ovule. Within, from the plant's perspective, the flower is a costly structure to build and maintain. The petals, the nectar, the fragrances, all require energy and resources, but the benefit, reliable transfer of pollen between individuals, is worth the cost because sexual reproduction shuffles genetic combinations in ways that can produce offspring better suited to varying conditions. The shapes and colors of flowers are often closely matched to the sensory capabilities and feeding behaviors of their pollinators. Flowers pollinated by bees tend to be blue, purple, or yellow colors that bees can see well and often have markings called nectar guides that are visible in the ultraviolet range of the spectrum, which bees can detect, but humans cannot. Flowers pollinated by butterflies are often red or orange and have narrow tubes suited to the long tongues of butterflies. Flowers pollinated by moths tend to be white or pale colored and strongly fragrant, since moths fly and feed at night when color is less useful, but scent carries across the dark. Some flowers are pollinated by birds, particularly hummingbirds, in the Americas, and these tend to be red, tubular, and rich in nectar. Others are pollinated by bats, by flies, by beetles, by small mammals. There are flowers that produce heat to attract cold-blooded insects seeking warmth. There are flowers that mimic the shape and scent of female insects, tricking male insects into attempting to mate with them and picking up pollen. In the process, there are flowers that produce fragrances chemically similar to rotting meat, attracting the flies and beetles that lay their eggs in carrion. Not all plants depend on animals for pollination. Many plants, including most grasses, many trees, and a number of other groups, are wind-pollinated. These plants tend to have small, inconspicuous flowers with no petals and no nectar. They produce enormous quantities of pollen, enough to drift in visible clouds from a grove of hazel trees on a calm morning, and rely on the statistical likelihood that some pollen will land on the pistil of another plant of the same species. The seasonal pollen clouds that provoke allergies in many people are the result of this strategy. Once pollen reaches the stigma of a compatible plant, it germinates, growing a tube that penetrates through the style of the pistil down to the ovule, through which the male genetic material travels to fertilize the egg cell. Within, the fertilized egg begins to divide and develop into an embryo. The surrounding tissues of the ovule harden and accumulate food reserves to become the seed. The ovary wall develops around the seed to become the fruit, a structure whose primary role is the protection and dispersal of the seeds within. The flower, having done its work, wilts and falls away. The fruit ripens, and inside the fruit, the seeds wait. The seed is, in many ways, the most concentrated form of potential in the living world. Enclosed in its coat, hard or soft, thick or paper-thin, depending on the species, is an embryo, a miniature plant, with the beginnings of root, stem, and leaf already laid down. Around the embryo is a supply of stored food, starch, oils, proteins that will sustain the seedling through the critical period between germination and the moment when it can photosynthesize enough to support itself, and over all of it, the seed coat, a structure designed to protect the embryo from drying out, from physical damage, from the digestive enzymes of animals And sometimes, from fire, seeds represent one of the most successful innovations in plant evolution. Before seeds, land plants reproduced through spores, small single-celled structures that had to land in a moist place and develop entirely on their own resources. Seeds changed the equation dramatically. A seed carries not just genetic information, but a prepackaged food supply and a protected embryo, ready to grow when conditions are right. The evolution of seeds, roughly 360 million years ago, appears to have been an important factor in the subsequent success of seed plants, conifers, cycads, ginkgos, and eventually flowering plants in colonizing a wide range of habitats. One of the most important properties of seeds is dormancy. A seed does not germinate the moment it forms. It waits. In many species, dormancy is maintained by chemical inhibitors within the seed that must be broken down before the embryo can begin to grow. In others, the seed coat must be weakened by water, by microbial action, or by the physical abrasion of passing through an animal's digestive system before germination can occur. Some seeds require a period of cold before they can germinate, an adaptation that prevents them from sprouting in autumn, only to be killed by the coming winter. Dormancy allows seeds to persist in the soil, sometimes for months, sometimes for years, occasionally for much longer, waiting for conditions that make successful germination likely. The collection of dormant seeds in the soil beneath a patch of vegetation is called the soil seed bank, and it represents a kind of living archive of the plant species that have grown in a location. Over time, research suggests that some seeds can remain viable in soil for decades, and a few exceptional cases of much longer dormancy have been documented, though these are rare and carefully studied. When conditions are right, when soil temperature, moisture, and sometimes light meet the germination requirements of a particular species, the seed begins to stir. Water enters through the seed coat, activating enzymes and triggering metabolic processes. The embryo begins to grow. The root emerges first, anchoring the seedling and beginning to absorb water and minerals. Then, the shoot pushes upward toward the light, carrying the first leaves, either folded within the seed or unfurling from the cotyledons. The seed leaves that provide the seedling with its initial food supply. But before germination can happen, the seed must travel, and seeds have evolved a spectacular range of mechanisms for moving through the environment. Some seeds ride the wind, the familiar papery wings of maple seeds, the ones that spin down through autumn air like tiny helicopters, are adaptations for wind dispersal. Dandelion seeds have their famous feathery plumes. Orchid seeds are so fine they drift in the air like dust. The key adaptation in wind-dispersed seeds is low mass, combined with structures that slow the rate of fall, maximizing the distance a seed can travel. On air currents, other seeds travel inside animals. Many plants enclose their seeds in fleshy, nutrient-rich fruits that are attractive to animals as food. The animal eats the fruit. The seeds pass through the digestive system unharmed and are deposited at some distance from the parent plant, often with a dose of fertilizer included. Some seeds travel on animals rather than through them. Paw prints, the burrs that catch on clothing and animal fur, are seeds equipped with hooks or barbs. Walk through a field of burdock in autumn, and you will have personally experienced this dispersal strategy. Water disperses the seeds of many plants. Coconuts are the most dramatic example. Large, buoyant, enclosed in a fibrous husk that keeps the seed fresh for months while it drifts across ocean currents. Some plants disperse their seeds by explosive mechanisms, building up tension in their seed pods as they dry until they burst, scattering seeds several meters in any direction. And in some ecosystems, seeds are cached by animals, squirrels, jays, rodents that store them as food reserves and then fail to retrieve them all, effectively planting them. Researchers believe that the large, nutrient-rich seeds of many oak and beech trees have co-evolved with the caching behavior of these animals. A seed, however small, contains within it the entire future of a plant, the patient potential of the seed, sitting in darkness, waiting for the particular combination of conditions that will allow it to begin, is one of botany's most enduring and quietly astonishing subjects. Stand at the edge of any woodland in the early morning, and you are standing at the threshold of a system, not a collection of individual organisms, not just this oak and that badger set and those ferns and that stream, but a system, an interlocking web of relationships in which material and energy flow from organism to organism in patterns that have evolved over millions of years. Plants are not merely present in this system. In most terrestrial ecosystems, they are its structural foundation. Begin with energy. Almost all of the energy flowing through land ecosystems enters through photosynthesis, through the conversion of sunlight into sugars by plants. Plants are what ecologists call primary producers. They produce the organic matter that all other organisms in the ecosystem ultimately consume. A deer eats grass. A wolf eats the deer. A bacterium decomposes the wolf. When it dies, in each transfer, energy stored originally in plant tissue is passed along and partially dissipated. Remove the plants, and the energy supply for the entire system collapses. The productivity of an ecosystem, the rate at which it produces organic matter, is therefore closely tied to the productivity of its plants. Tropical rainforests, with their high temperatures, abundant rainfall, and year-round sunlight, are among the most productive ecosystems on Earth, producing enormous quantities of plant biomass every year. Tundra and desert ecosystems, constrained by cold, drought, or both, are far less productive. The gradient of plant productivity across the globe is reflected in the gradient of animal diversity. More plant productivity supports more animal life at every level of the food web. But plants do not just provide energy. They shape the physical environment that other organisms live in. A forest is not just a collection of trees. It is a structure, a three-dimensional space, divided into layers, each with different conditions of light, temperature, humidity, and wind. The canopy, the understory, the shrub layer, the ground layer, each is a distinct habitat, occupied by organisms, adapted to its particular conditions. The forest creates these layers. The trees that compose the canopy determine how much light reaches the ground, how much rain is intercepted before it reaches the soil, how much wind penetrates the interior. Remove the canopy, and the structure collapses, along with the specialized communities of organisms that inhabited each layer. Plants also shape the soil. Root systems stabilize soil against erosion by rain and wind. Plant litter fallen leaves, dead stems, shed bark, accumulates on the soil surface and is decomposed by fungi and bacteria into humus, the dark organic component of fertile soil. The organic content of soil determines its water-holding capacity, nutrient content, texture, and structure. Forest soils, built up over centuries by the slow accumulation of plant litter, are profoundly different from the soils of a freshly cleared field. Deeper, darker, richer, more alive, the influence of plants on the water cycle is substantial. Forests intercept rainfall, slowing the rate at which water reaches the ground. Plant roots take up water from deep soil layers and release it back to the atmosphere through transpiration. In large forested regions, the volume of water cycled through trees by transpiration is enormous, large enough, researchers believe, to influence regional rainfall patterns. The removal of large forests has been associated, in some studies, with measurable changes in local climate conditions, including reduced rainfall and increased temperature variability. Plants also influence the atmosphere. Photosynthesis removes carbon dioxide from the atmosphere and stores it in plant tissues and soils. This storage of carbon in vegetation and soils represents a significant component of the global carbon cycle. Estimates suggest that plants and soils currently hold several times more carbon than is present in the atmosphere, and the balance between plant uptake and release through respiration and decay is a major factor in global carbon dynamics. Scientists continue to study these processes and refine their estimates of how different types of vegetation contribute to the global carbon budget. Grasslands, wetlands, and coastal ecosystems each play distinct roles in the functioning of Earth's biosphere. Wetlands, marshes, bogs, swamps, and fens are particularly important for water storage and filtration. The dense plant communities of wetlands slow the movement of water and trap sediments and excess nutrients, acting as natural filters for water moving toward rivers and lakes. Coastal salt marshes and mangroves provide nursery habitats for many fish species. Grasslands, covering large areas of the continents, support vast communities of grazing animals and store significant quantities of carbon in their deep, organic-rich soils. The relationship between plants and the ecosystems they inhabit is not static. It changes through time in a process ecologists call succession. A bare patch of ground, freshly exposed rock, a field left fallow, a forest clearing, does not remain bare. Pioneer species, typically small, fast-growing plants with wind-dispersed seeds, are the first to colonize. They modify the environment, accumulating organic matter, stabilizing the soil, creating shade. This makes conditions suitable for slower-growing, larger species that could not have established on bare ground. Over time, through a sequence of changing plant communities, a bare patch of ground may develop into a complex, layered forest. This process of succession reminds us that the plant communities we see around us are not permanent features of the landscape. They're stages in an ongoing process of ecological development, shaped by the plants that have grown there before, and by the conditions, climate, soil, disturbance, that govern which plants can grow there now. The story of human civilization is, in very large part, a story about plants. From the earliest evidence of human ancestors using plant foods in ancient Africa to the global agricultural systems that feed 8 billion people today. Plants have been at the center of human life in ways that are easy to overlook, precisely because they are so fundamental. The dietary foundation of human existence is almost entirely botanical: wheat, rice, corn, barley, oats, sorghum, millet, the great cereal crops that have sustained civilizations on every inhabited continent are all grasses whose seeds are harvested for food: potato, sweet potato, cassava, yam, the root and tuber crops that have fed populations from the Andes to sub-Saharan Africa to Southeast Asia are all plants. Legumes, beans, lentils, chickpeas, soybeans provide protein across an extraordinary range of cuisines and cultures. Fruits, vegetables, nuts, oils, almost all of it comes from plants. Before agriculture, human populations sustained themselves through the gathering of wild plant foods, supplemented by hunting. The gathering component of this hunter-gatherer lifestyle required and generated detailed botanical knowledge of which plants grew where, when different plant foods were available through the seasons, how to process plants that were toxic in raw form, how to store plant foods for periods when fresh food was unavailable. This was not casual knowledge. It was deep, accumulated, carefully transmitted, a science of plants, even if it was not yet called botany. The transition to agriculture, the deliberate cultivation of plant crops, appears to have occurred independently in several parts of the world, beginning roughly 10,000 years ago, during a period of climatic stabilization. At the end of the last glacial period, in the Fertile Crescent of the Middle East, wheat and barley were domesticated from wild grasses. In China, rice and millet cultivation developed independently. In the Americas, corn was developed from a wild grass called teosinte through a process of artificial selection so dramatic that the resulting plant, corn, looks almost nothing like its wild ancestor. Domestication, the process by which wild plants are gradually modified through human selection to have characteristics more useful for agriculture, is one of the most profound examples of directed evolutionary change. Over generations, early farmers selected the seeds they planted from plants that had desirable traits: larger seeds, seeds that stayed on the plant instead of scattering when ripe, plants that grew at uniform heights, plants that matured at the same time. Gradually, through centuries of unconscious and later conscious selection, the wild ancestors of our crops were transformed into the domesticated varieties we recognize today. The changes wrought by domestication were not just agricultural. The development of reliable food surpluses, made possible by agriculture, allowed human populations to grow, to settle in fixed locations, to develop specialization of labor, to build cities. The civilizations of ancient Mesopotamia, Egypt, China, India, and Mesoamerica were all built on the foundation of agricultural surpluses, on the reliable productivity of domesticated plant crops. Beyond food, plants have provided humanity with an extraordinary range of other materials. Timber, the wood of trees, has been used for construction, for fuel, for tools, for ships, for furniture, across all of recorded history and prehistory. Without timber, the great forests of northern Europe could not have been cleared. The great cathedrals of the medieval period could not have been built. The ships that carried exploration and trade across the oceans could not have been constructed. Plant fibers have clothed human beings. For millennia, cotton, the fiber surrounding the seeds of a tropical plant, is still among the most widely used textile fibers in the world. Flax provided linen, the fabric of ancient Egypt. Hemp, jute, sisal, the plant world has provided a remarkable diversity of fiber materials, each with different properties, suited to different uses. The medical traditions of virtually every human culture draw extensively on plants. The history of medicine is, in large part, a history of botanical medicine, of healers learning which plant compounds could reduce fever, ease pain, fight infection, stimulate the heart, or calm anxiety. Many of the most important drugs in modern medicine trace their origins to plant compounds. Willow, whose bark compounds led to aspirin; opium poppy, source of morphine; cinchona, foxglove. Yew, the systematic investigation of plant-derived medicines continues today, and research scientists continue to screen plant compounds for biological activity. Plants also provide the world's populations with beverages. Tea plant, tea from the dried leaves of a plant native to East Asia. Coffee plant, coffee from the roasted seeds of a tropical shrub. Cacao tree, cocoa from the fermented seeds of the cacao tree. Fermented plant products have provided alcohol in every culture. Wine from grapes, beer from barley and wheat, spirits distilled from sugarcane, corn, potatoes, and dozens of other plant materials. The dependence of human civilization on plant life is so deep and so pervasive that it can be difficult to fully comprehend. Remove plants from the human world, and the structure of civilization, its food systems, its materials, its medicines, its landscapes, its cultures, would be unrecognizable. The science of plants today looks very different from the science practiced by Theophrastus in ancient Greece, or even by the great Victorian botanists who cataloged the world's plant diversity. In the 19th century, modern botany has been transformed by new tools: molecular biology, genomics, imaging technology, computational analysis, tools that have opened up levels of plant organization entirely invisible to earlier researchers. Perhaps the most significant transformation has been the development of plant genetics and genomics, the ability to read the DNA sequence of a plant, to identify the specific genes it carries, and to study how those genes are expressed in different tissues and under different conditions, has given botanists tools of extraordinary power. Scientists can now identify the genetic basis of traits like drought tolerance, disease resistance, crop yield, and nutritional content, opening up new approaches to crop improvement. The first complete plant genome to be sequenced was that of Arabidopsis thaliana, a small flowering plant in the mustard family, published in the year 2000. Arabidopsis has become the model organism of plant biology used in laboratories around the world as a tool for studying fundamental plant processes. Its small genome, short generation time, and ease of genetic manipulation have made it invaluable for dissecting the molecular mechanisms that underlie everything from light sensing to disease resistance to the timing of flowering. Since the sequencing of the Arabidopsis genome, the genomes of hundreds of plant species have been sequenced, including virtually all of the major crop species: the genome of rice, the genome of maize, the genome of wheat. These have been analyzed in remarkable detail, and the information gained is being used to guide plant breeding programs aimed at improving crop productivity and resilience. Modern plant breeding has been transformed by molecular tools. Traditional plant breeding involved crossing plants with desirable traits and selecting among the offspring, a process that could take many years per generation, and that moved desired traits into new varieties only slowly. Molecular markers, specific sequences of DNA associated with desirable traits, allow breeders to select plants carrying the desired genes at the seedling stage without waiting for the plants to mature and express the relevant traits. This accelerates the breeding process considerably. More recently, gene editing technologies, particularly CRISPR-Cas9, have provided plant scientists with tools to make precise, targeted changes to plant genomes. These technologies allow researchers to modify specific genes without introducing DNA from other species, a distinction that is relevant both scientifically and in terms of the regulatory and social questions that surround genetically modified crops. Research using these tools is ongoing across many areas of plant science, from improving disease resistance to developing crops with enhanced nutritional profiles. The scientific, ethical, and regulatory dimensions of these technologies continue to be actively discussed by researchers, regulators, and the public. Beyond genetics, modern plant science has made significant advances in understanding the molecular biology of plant development, how a fertilized egg cell develops into a complete plant, how leaves, flowers, and roots are specified and shaped, how the timing of key developmental events is regulated, the molecular mechanisms governing flowering time, for instance, have been worked out in considerable detail over the past few decades, revealing a complex network of genes and environmental signals that integrate information about day length, temperature, and the plant's own internal state to determine when it is appropriate to flower. Greenhouse and controlled environment agriculture have expanded the possibilities of food production in environments otherwise unsuitable for growing. Modern greenhouse systems, using artificial lighting, hydroponics, the cultivation of plants in nutrient solutions without soil, and precisely controlled temperature and humidity, can produce crops year-round in climates where outdoor growing is seasonal or impossible. These systems are energy-intensive, but research continues to improve their efficiency, and in contexts where land is limited or climate is unfavorable, they offer an important supplement to field agriculture. The science of plant physiology, the study of how plants function, has continued to advance, revealing subtleties and mechanisms that earlier generations of botanists could not have imagined. The molecular basis of photosynthesis has been worked out in remarkable detail, the signaling pathways through which plants perceive and respond to light, water stress, pathogens, and other environmental factors are being systematically elucidated. The mechanisms by which plants produce secondary metabolites, the thousands of chemical compounds that give plants their flavors, fragrances, colors, and medicinal properties, are being studied with increasing precision. Modern botany is a rich and active field, drawing on an ever wider range of tools and approaches to address questions about plants that have fascinated scientists and observers for millennia. And at its core, it remains what it has always been: careful, attentive, patient observation of the extraordinary variety and ingenuity of plant life. Plants are not passive recipients of the conditions they encounter. They are, within the limits of their fixed positions, remarkably responsive organisms, capable of adjusting their physiology, their growth patterns, and even their chemical profiles in response to changing conditions in their environment. Understanding how plants respond to environmental variation, including long-term changes in climate, soil conditions, and atmospheric composition, is one of the important areas of current plant science research. Temperature is one of the most fundamental environmental variables affecting plant life. Every plant species has a characteristic range of temperatures within which it can function. Below a minimum, enzyme activity slows. Cells may be damaged by freezing. Above a maximum, proteins begin to denature. Metabolic processes break down. Within this range, temperature affects photosynthetic rates, growth rates, developmental timing, and many other physiological processes. Plants native to cold climates have evolved remarkable adaptations for surviving frost. Arctic and alpine plants are often low-growing, staying below the level of the wind and sometimes snow, which provides insulation. Many develop chemical changes in their cell membranes during autumn that reduce freezing damage. Some produce antifreeze proteins. Others can survive complete desiccation at very low temperatures. Rehydrating when conditions improve. Understanding the molecular basis of frost tolerance is an active area of research with potential applications in breeding cold-tolerant crops. Water availability is another critical variable. Plants native to arid environments have evolved diverse strategies for managing water scarcity. The thick, waxy cuticles and reduced surface area of succulent plants, the deep taproots of some desert shrubs that can reach water tables far below the surface, the ability of many desert annuals to compress their entire life cycle into the brief wet season, remaining dormant as seeds through the long dry periods between. Current evidence indicates that plants regulate their water use at the molecular level through complex signaling systems involving hormones, particularly abscisic acid, a hormone produced by plant cells under water stress, which triggers stomatal closure, helping the plant conserve water beyond temperature and water. Plants respond to a wide range of other environmental variables. Light is among the most important. The quality and quantity of light affects plant development, growth form, and the allocation of resources between roots, stems, and leaves. Plants in deeply shaded environments often produce larger, thinner leaves with more chloroplasts per unit area. Adaptations designed to capture as much light as possible under low-light conditions. Plants in high-light environments may produce smaller, thicker leaves, sometimes with reflective surfaces that protect them from excess sunlight. Nutrient availability shapes plant growth in fundamental ways. In nutrient-poor soils, plants often allocate more resources to root growth, exploring larger volumes of soil to meet their nutritional needs. They may also form extensive associations with fungi called mycorrhiza, which help plants absorb nutrients more efficiently. Some plants have evolved an extraordinary solution to obtaining nitrogen, an essential nutrient often scarce in soils. Members of the family Fabaceae, which includes beans, clover, and alfalfa, form nodules on their roots. Inside these nodules, live nitrogen-fixing bacteria, which convert atmospheric nitrogen into forms the plant can use. Plants also respond to biological environmental factors, herbivores, pathogens, and competing plants. When plant tissue is damaged by chewing insects, Many species release volatile chemical compounds into the air. These chemicals can act as signals to neighboring plants, priming them to activate their own defenses. Plants also produce a wide range of defensive compounds, bitter tannins, toxic alkaloids, and physical deterrents, like thorns or hair-like structures called trichomes. These defenses reduce the damage inflicted by herbivores and pathogens. Research suggests these responses can be highly specific, with different chemical responses triggered by different types of attack. Long-term shifts in environmental conditions, climate, soil chemistry, and species composition drive evolutionary responses. Over generations, the distribution of plant species across the landscape reflects, among other things, the history of climatic change over geological timescales. During glacial periods, when temperatures were lower and vast ice sheets covered much of the northern continents, the ranges of many plants contracted toward lower latitudes and elevations. When glaciers retreated and temperatures rose, plant populations expanded, tracking shifting climate zones and recolonizing previously glaciated landscapes. Plant ecologists have reconstructed these migrations through fossil pollen records preserved in lake sediments and peat bogs. The capacity of plant populations to respond to environmental change through individual plasticity, through migration, and over longer timescales, through evolution, is a subject of considerable scientific interest. How quickly plant populations can track changing conditions and whether rates of change may outpace their capacity to respond are questions that ecologists and evolutionary biologists continue to study carefully. There is a tree in the mountains of eastern California that is considered to be among the oldest living organisms on Earth. It is a bristlecone pine, and researchers estimate its age based on the rings in its trunk and the matching of those rings to a long series of historical climate records at over 4,000 years. It was alive when the ancient Egyptians were building the pyramids. It was alive when Homer composed the Iliad. It was alive before the Buddha was born. It looks from a distance like a gnarled, weathered, largely dead tree. Most of its trunk stripped bare by centuries of wind and weather. Only a narrow strip of living bark still connecting its roots to a few green branches at the top. It is not magnificent in the way that a tall, symmetrical, full-crowned tree is magnificent. It is magnificent in the way that endurance is magnificent, in the way that something that has persisted through everything time could throw at it, and is still alive, is magnificent. Trees are the longest-lived individual organisms most of us will ever encounter. A mature oak tree in an English countryside may be 300, 400 years old. A giant sequoia in California may be 2,000 years old or more. A Scots pine in a remote Scottish glen may have been growing for centuries, its rings recording the history of cold winters and warm summers. In the width of the annual layers laid down within its trunk, the way trees grow is the key to understanding their longevity. Unlike animals, trees do not grow by the uniform enlargement of a body that is laid down in its final form during development. Trees grow by addition, by continuously adding new tissue to their growing tips and their trunks, year after year, without limit except that imposed by disease, damage, or environmental stress. Each year, a layer of new cells is added to the outside of a tree trunk, just beneath the bark. These cells, produced by a thin layer of dividing cells called the vascular cambium, develop into new xylem on the inside and new phloem on the outside. The xylem, added in spring when conditions for growth are favorable and water is abundant, tends to have large thin-walled cells. It appears pale in a cross-section of the trunk. The xylem, added later in the growing season, as conditions become less favorable, has smaller, thicker-walled cells and appears darker. This alternation of pale and dark bands is what produces tree rings, the annual growth rings that have been used by dendrochronologists, scientists who study tree rings, to reconstruct centuries of climate history. In tropical trees, where seasons are less pronounced, growth rings may be less distinct or absent. In trees growing in strongly seasonal climates, cold winters, warm summers, wet seasons and dry seasons, ring width and density can vary dramatically from year to year, reflecting the quality of growing conditions. Wide rings indicate favorable growing conditions. Sufficient water, adequate temperatures, long growing seasons. Narrow rings indicate stress, drought, cold, disease, damage. Reading these rings requires considerable skill and an understanding of the ecology of the specific tree species, but the information they contain is extraordinarily valuable. The oldest sections of a tree trunk, the innermost rings, are no longer involved in the transport of water. The central wood of old trees is typically dead, non-conducting tissue, called heartwood, which provides structural strength, but no longer participates actively in the tree's physiology. The conducting xylem, the sapwood, is confined to the outer rings, just inside the vascular cambium. A very old tree carries within it a record of its own history. The heartwood at the center represents the earliest years of its growth, laid down when it was young, and perhaps small. The outermost rings represent the current year. Trees in a forest community are not isolated individuals. Their root systems are intermingled and often interconnected through mycorrhizal networks. Their canopies interact in complex ways, competing for light, creating mutual shading, affecting the climate beneath them. Scientists have studied how trees in a forest respond to the death or decline of neighbors, how gaps in the canopy created by fallen trees allow the growth of understory trees, and how species composition in a forest changes over decades. As some species grow faster and overtop others, the dynamics of old-growth forests, forests that have developed over centuries without major disturbance, are particularly complex. Old-growth forests tend to have a multi-layered canopy, with trees of many different ages and sizes, a higher proportion of dead and decaying wood than younger forests, and a distinctive suite of species adapted to the particular conditions they provide. Understanding how these communities develop and function is an important area of forest ecology research. The oldest trees are survivors. They have persisted through droughts, through windstorms, through insect outbreaks, through fires, through centuries of climate variation. Their persistence tells us something about the resilience of plant life, about the capacity of organisms that cannot move, cannot flee, cannot fight, in any conventional sense, to endure through almost anything the world can offer. One of the most persistent assumptions about plants is that they are simple, that compared to the rich inner life of animals, the behaviors, the communications, the social structures, plants are merely passive, mechanistic, reacting to stimuli, without any of the elaboration that characterizes animal responses. Modern plant science is painting a somewhat more complicated picture. It is important to be careful here. Plants do not have brains, nerves, or anything structurally analogous to a nervous system. They do not have consciousness in any sense that current science can detect or define. When plant biologists speak of plant signaling, plant communication, and plant responses, they are speaking of biochemical processes, the production and reception of chemical signals, the activation of gene networks, the physical and physiological changes these processes produce. There is no need to invoke anything beyond chemistry and physics to explain what plants do And what plants do, it turns out, is considerably more than just grow toward light and absorb water. Plants can detect and respond to a remarkable range of stimuli. They sense light, not just its presence and absence, but its quality. The ratio of red to far red light tells a plant whether it is in open sunlight or shaded by other vegetation. And this information influences development and growth patterns. Plants sense gravity. Their roots grow downward, and their shoots grow upward in response to gravitational cues, mediated by specialized cells containing starch granules that settle to the bottom of the cell under gravity. Plants sense touch. Climbing plants extend tendrils that curl rapidly around support structures. When they make contact, a response mediated by changes in cell turgor pressure. When plants are damaged by herbivores or pathogens, they mount chemical defenses. In many species, damage to one part of the plant leads to defense responses throughout the plant. The production of bitter or toxic compounds, the reinforcement of cell walls, the activation of immune-like responses, mediated by chemical signals traveling through the plant's vascular system, and sometimes through the air as volatile compounds. The possibility that these airborne volatile signals can affect neighboring plants, plants that share the same space but are not physically connected, has been the subject of considerable research and some controversy. Research suggests that some plant species do produce volatile compounds when damaged by herbivores, and that neighboring plants of the same or different species can respond to these volatiles by upregulating their own defenses. The precise ecological significance of this phenomenon, how important it is in natural conditions, how far signals carry, how consistently responses occur, continues to be studied and debated in the scientific literature. Researchers are careful to distinguish between results obtained under controlled laboratory conditions and what happens in complex field situations. Below the soil, as noted earlier, the mycorrhizal networks that connect plant roots to fungi appear to allow the movement of nutrients between connected plants. Whether and to what extent carbon compounds and nutrient signals move through these networks in ways that represent something like communication between plants, as opposed to simply reflecting the flow of resources through shared fungal tissue, is a question that researchers continue to investigate carefully. The data are intriguing, the mechanisms are being studied, and cautious scientific phrasing is appropriate. Current evidence suggests that these networks can facilitate resource transfers between plants, but the interpretation of what this means for plant ecology and behavior is still developing. Plants also have complex internal signaling systems that coordinate development and physiology across the plant body. Plant hormones, chemical signals produced in one part of the plant and transported to other parts, where they have effects, play central roles in this coordination. Auxin, one of the most studied plant hormones, is produced at growing tips and moves downward through the plant, influencing cell elongation, branching, and root development. It is the hormone primarily responsible for phototropism, the bending of plant shoots toward light, which it achieves by causing cells on the shadowed side of the shoot to elongate faster than those on the illuminated side. Other plant hormones include gibberellins, which promote stem elongation and seed germination, cytokinins, which promote cell division and delay aging in leaves, ethylene, a gas that promotes fruit ripening and is involved in the responses to flooding, wounding, and other stresses, and abscisic acid, which promotes dormancy and stomatal closure under water stress. The interactions among these signaling molecules and between them and environmental signals constitute a network of extraordinary complexity that regulates almost every aspect of plant development and physiology. The more carefully plant scientists look at the biochemical and molecular organization of plants, the more complex the picture becomes, not more mysterious. The mechanisms are amenable to scientific investigation, and they are being investigated with increasing rigor. But more surprising, in the best sense of the word, the plant that sits quietly in a pot on a windowsill is not doing nothing. It is monitoring light quality and duration. It is regulating water use through its stomata. It is producing secondary metabolites. It is growing in response to 100 different signals. It is, in its quiet and unhurried way, an information processing system of considerable sophistication. Plant science today stands at an interesting junction of challenges and opportunities. On one hand, the tools available to plant scientists are more powerful than at any previous point in history. Genomic sequencing is fast and cheap enough to be routine. Imaging technologies allow researchers to follow the development of individual cells in living plants in real time. Computational tools allow the analysis of datasets of previously unmanageable size. These capabilities are accelerating the pace of discovery in every area of plant biology. On the other hand, the questions that plant science is being asked to help address are increasingly urgent and complex. How do we feed a growing global population while maintaining the natural systems that support agriculture? How do we develop crops that can tolerate the range of temperature and water conditions that farmers may face in coming decades? How do we understand and protect the plant diversity that represents millions of years of evolutionary experimentation and that underpins the functioning of ecosystems worldwide? These questions do not have simple answers, but plant science is contributing to the development of answers in several important ways. In plant genetics, the combination of detailed genomic knowledge and gene editing tools is opening up new possibilities for crop improvement. Researchers are working on developing varieties of major crops with greater tolerance to drought and heat, improved resistance to major diseases and pests, and enhanced nutritional profiles. Some of this work involves the introduction of genes from wild relatives of crop species, plants that evolved under the selection pressures of variable environments, and that carry genetic variants for stress tolerance that were lost during the domestication process. Current research is exploring how to transfer these useful variants back into high-yielding cultivated varieties. The science of plant microbe interactions, the study of how plants interact with the communities of bacteria and fungi in the soil and on plant surfaces, is revealing new opportunities. Research suggests that plant growth and health are influenced in significant ways by the composition of the microbial community associated with plant roots. Some researchers are exploring whether it is possible to design or manage these microbial communities, what is now called the plant microbiome, to improve plant growth and reduce dependence on chemical fertilizers and pesticides. This is a relatively young field, and the results are promising, but require further development before they can be broadly applied in agriculture. In forest science, research is focused on understanding how different types of forests respond to changing conditions, what makes some tree species more resilient than others, and how forest management practices can promote the development of diverse, resilient forest communities. The science of assisted migration, deliberately moving plant populations to locations where climate projections suggest they may be better suited than current populations, is being explored as a potential tool for maintaining plant diversity and forest function in the face of rapidly changing conditions. This is a complex area with both scientific uncertainties and practical and ethical dimensions that researchers and managers are working through carefully. Conservation botany, the science of understanding and protecting plant diversity, is another important frontier. Researchers estimate that a significant proportion of plant species face threats to their survival, though the precise figures are difficult to determine, given how poorly some groups are known scientifically. Seed banks, collections of seeds from thousands of plant species, stored at low temperatures, represent one approach to preserving plant genetic diversity. The Svalbard Global Seed Vault, located in the Arctic, holds copies of seed collections from around the world as a safeguard against the loss of crop and plant diversity. Botanical gardens around the world maintain living collections of plant species, contributing to both conservation and the scientific study of plant biology. Basic plant science, the investigation of fundamental mechanisms of plant biology, continues to advance rapidly. Researchers are working to understand the molecular basis of plant aging, the mechanisms by which plants sense and respond to light signals, the genetic basis of the extraordinary chemical diversity of plant secondary metabolites, and the evolutionary processes that have generated the immense diversity of plant form and function. Each of these areas is generating new knowledge that has both intrinsic scientific value and potential practical applications. The future of plant science will be shaped, as all science is, by the questions that researchers ask and the tools they have available to answer them. Simple science explanations for beginners often present science as a collection of known facts. But the most exciting part of any science is the frontier, the boundary between what is known and what is not yet understood. Botany has a rich and active frontier, and the work of understanding plants is far from finished. Return for a moment to the forest at dawn. The light has shifted since we first arrived here. The deep blue of early morning has given way to the pale gold of an hour after sunrise. The mist has lifted. The first insects are beginning to move through the understory. Somewhere in the canopy above, a leaf is opening its stomata in response to the increasing light, beginning the day's work of gas exchange and photosynthesis. Below the surface of the soil, root hair cells are absorbing water from the film that coats the soil particles around them, and that water is beginning its slow upward journey through the xylem of 1,000 roots and stems and branches. The seed we noticed at the beginning of our journey, that small, still object resting in the dark below the leaf litter, has perhaps shifted slightly. Moisture has penetrated its coat. Enzymes have begun to stir. The chemical inhibitors that have maintained dormancy are slowly breaking down. Perhaps this morning will not be the morning it germinates, but the conditions are moving in the right direction. There is no urgency here, no ticking clock, no deadline. Plants do not experience time the way we do, and they do not need to. A seed that waits 10 years for the right combination of conditions before germinating has not failed. It has succeeded. It has survived long enough to find a moment when its chances of establishment are high. This patience is perhaps one of the qualities we most need to appreciate in order to truly understand plants. We are accustomed to biological systems that respond at the pace of animal life, heartbeats and breaths and movements measurable in seconds. Plants operate on a different clock. Cells dividing, roots extending, seasons cycling, rings accumulating, decades passing. The time scale on which plant life makes sense is longer than the time scale on which we usually attend to the world. But the science we have explored in this documentary shows us that slowness is not the same as simplicity. Within the unhurried rhythms of plant life, there is a level of biochemical and structural complexity that scientists are still, in many places, only beginning to understand. The molecular machinery of photosynthesis, the intricate signaling networks that coordinate plant development, the underground web of relationships between roots and fungi and bacteria, the chemical conversations between plants and their pollinators, their herbivores, their pathogens, the genetic systems that encode the extraordinary diversity of plant form across the world tonight. While most of us sleep, plants are doing what they have done for hundreds of millions of years, photosynthesizing, transporting water, growing by fractions of a millimeter, exchanging chemical signals with the soil community around their roots, preparing to flower, or to fruit, or to set seed, doing in darkness and in silence the continuous work that makes the living world possible. A forest breathes across the night, oxygen and carbon dioxide, moving through millions of open stomata. A wetland marsh holds water against the dry season, its dense roots and thick organic soils, absorbing the excess of the rains. A single dandelion on a suburban pavement pushes its taproot through a crack in the concrete, demonstrating with perfect equanimity that the capacity of plant life to find and use any available space is remarkable, patient, and persistent. The plant world does not make demands. It does not announce itself. It simply grows, leaf by leaf, ring by ring, root by root, building the structures and doing the chemistry that everything else depends on. The science of botany is, in the end, the science of learning to pay attention to the leaf on a branch, to the root in the soil, to the seed in its coat, to the light captured, the water moved, the sugar formed, the gas exchanged, to the slow, continuous, invisible work of organisms that have been here far longer than we have, and that have shaped the world into the form in which we found it, across forests, fields, and mountains, across wetlands and grasslands, and the margins of every lake and river. The plant world continues its quiet work, turning sunlight into life, growing leaf by leaf, season by season, year by year, always slowly, always patiently, always in its profound and unhurried way, building the world